It's my pleasure to introduce Professor Lee Gan from Weill Cornell Medical College, and she's going to talk today to us about maladaptive microglia and how they drive tauopathy. So, Professor Gan.
Great. So thanks so much, Li Hui and the organizers for this incredible opportunity. And I'm really glad to share our recent research on microglia. And Beth has set up such a beautiful background on microglia. So today, as Ravi said, I will be talking about microglia in tauopathy, and in particular in the context of cognitive resilience. So if we could describe the progression of Alzheimer's disease in a trilogy, we can put it into three overlapping chapters. So that for the chapter one could describe as the start with proteostasis failure, and in the case of Alzheimer's disease, it would be amyloid and tau. And then that is followed by the innate immune response in the form of microglia reaction, other glia reaction, as well as vascular changes. And at the end, it invariably ends with a decline, the sad chapter 3, the decline of cognitive function as well as dementia. So what triggers the innate immune response is often the proteostasis failure. And as I mentioned in the AD, it's amyloid and tau, and it often starts at very regional and then spread to the other brain regions. And one need to be, one thing that needs to be emphasized is that even though amyloid and tau are the major pathology, the AD brains can often seen with inclusions of alpha-synuclein in the form of Lewy body, as well as TDP-43 that pathology observed in some part of the some AD brains as well. So another observations that have been made is that our brains are actually quite resilient because the cognitive dysfunction don't occur until many years after the proteostasis failure. And for the example of amyloid deposition precedes the cognitive deficit many years before the onset of MCI. So suggesting that we have a period of resilience against amyloid and tau pathology, and this resilience is gradually declining, and that is the reason that our brain loses function in the dementia stage. So what drives this loss of cognitive resilience against amyloid and tau? So today I will give you our working hypothesis is that we think that maladaptive microglia are the drivers of this loss of cognitive resilience. So why we say that? First, that let's think about what is microglia response, which is brain's innate immune response. Its function is to defend brain's functional and structural homeostasis. So the reason our brain are resilient to those changes is because we have defenders that are built in to help defend the brain to return to functional and structural homeostasis when they are being perturbed, such as with deposition of amyloid and tau. And another, most important clue that this could be the reason for a loss of cognitive resilience is because the most or vast majority of the risk genes that associate with late onset sporadic AD are the genes expressed in microglia. So suggesting that the changes, genetic SNPs on the microglia are contributing to a loss of cognitive resilience. So I wanted to illustrate this working model using two small vignettes. The first vignette, I want to describe a published work that we show that NF-kappa B activation drives tau spread and toxicity. It's published, so it's going to be really short. I will spend most of the time talking about the unpublished work of vignette 2, which I will discuss a working model how cGAS-STING interferon antiviral pathway could be driving the loss of cognitive resilience. So I will use tau pathology or tauopathy as illustrating the proteostasis failure in the AD as well as other primary tauopathies. So tau accumulation or accumulation of tau aggregates is closely associated with neurodegeneration, and in AD, it is associated with neurodegeneration more closely than amyloid plaque deposition. And other primary tauopathies, including familiar frontotemporal dementia tauopathy and other PSP, CBD, PIC disease, as well as CTE, are primary tauopathies that are characterized with accumulation of tau aggregates. So to model tauopathy, we used a very well-established model called PS19 that expressed, it's a transgenic mouse model expressing mutant form of tau, carry P301S mutation, a mutation that causes familiar form of ATD. We then isolated microglia and performed RNA-seq. And the most, if you do a pathway analysis of the upstream regulators, one of the top upstream regulators is IKB-KB, which is a kinase that is critical for the phosphorylation of I-kappa B alpha, as well as translocation of P65 and P50. So it is at the critical, it is a critical enzyme for canonical kappa B activation. So to dissect what does kappa B signaling do in microglia in the tauopathy mouse models, we use two approaches. We either selectively delete it in adult microglia, or we constitutively activate it in adult microglia. And they are using the CX3CR1 CRE-ERT model, and by either remove the IKB-KB from microglia or remove the stop from the constitutively active form of IKB-KB. We then look at how does it affect the spread of tau inclusions. So to do that, we inoculated tau fibrils into one side of the hippocampus of the young PS19 mice that lacking kappa B, and then examined what are the number of tau inclusions either in the ipsilateral side or in the contralateral side to examine both seeding as well as spread. And you can see here, like within a month, you can see a widespread of tau inclusions labeled with MC1 antibody, a confirmation-specific antibody, and examine the changes of the number of tau inclusions. And you can see that tau inactivating microglia with kappa B leads to a significant reduction of tau inclusions, especially in the ipsilateral side, but also at the same trend in the contralateral side, showing that inactivating kappa B reduces tau seeding and suggesting that activation of kappa B enhance tau seeding. And that is indeed true if we now constitutively active kappa B using this constitutively active form of IKB-KB in adult microglia, we see that the amount of tau inclusions is significantly elevated in the ipsilateral side, as well as to some extent in the contralateral side. So this tells us that not only the tau accumulation can drive kappa B activation, kappa B activation can further feed into the tau proteostasis failure. We have also shown that kappa B activation is underlying the spatial learning and memory deficit, because deleting microglia with kappa B can restore that. So, so this is showing the critical role of NF-kappa B. Another important signaling that is related to NF-kappa B is the interferon pathway. So that is the second part of my talk. The interferon pathway that is linked to NF-kappa B, so they have a very extensive crosstalk, as I will mention in the next slide. It has been shown to be highly enriched or activated in the human AD brains. So this shows this work from Wei's house and then Wei's lab a couple of years ago, look at over 178 brains and looked at the transcription network, transcriptional signature, and you can see that in the demented cases, the interferon transcriptional stimulated genes are highly elevated. And both NF-kappa B as well as interferon pathways can be activated through a pathway that involves STING. The STING pathway, the upstream of STING pathway, one of the major activator of STING pathway is cGAS. So cGAS is a cyclic GMP synthase that is a major sensor, not the only sensor, but one of the major sensors for double-stranded DNA in the cytosol. And that can, with the double-stranded DNA in the cytosol that is sensed as a danger signal or viral signal, infection signal, it will activate STING and leads to either activation of NF-kappa B as well as IRF3, and that leads to type 1 interferon response as well as some cytokines. So what we have with collaborative work with Joe Uduccio, a former postdoc, set up AMI, a cover postdoc, and graduate student Gloria Wang, we have been dissecting this cGAS interferon pathway and in relationship with cognitive resilience. So what Joe have found is that if he took the bulk seq, meaning not the microglia isolated, but the tissue from hippocampus of PS19 mice, and look at the hallmark of the hallmark pathway changes of the bulk RNA transcriptome. And what he see is interferon gamma response and interferon alpha response that are very, very highly upregulated. And in human microglia, what the doc has been looking at is to see, is this interferon response related to cGAS-STING pathway? So what she has been doing is to treat the human iPSC-derived microglia with tau fibrils and look at the phospho-STING and to see if phospho-STING is increased. And you can see there's a dramatic increase of phospho-STING in the human microglia treated with tau fibrils. And this increase of phospho-STING can be abolished by either cGAS inhibitor that's synthesized based on this publication, as well as the STING inhibitor H151. So showing that the cGAS-STING activation underlies the tau-mediated phospho-STING increase. So we looked at how the cGAS get activated by tau. So one of the key components is double-stranded DNA. And this double-stranded DNA could come from genomic instability, could also come from mitochondrial damage. So the first question that Sadaf asked is, do we actually see double-stranded DNA in the cytosol with tau stimulation? And indeed, she did. So by separating the cytosol from the rest of the organelles and quantify the double-stranded DNA normalized with nuclear DNA, and she found there's a significant increase of cytosolic DNA with tau treated microglia. So then where does it come from? So to address this question, depleted mitochondrial DNA with two approaches. Either use DDC or theobromide that leads to mitochondrial DNA mutation. And you can see the reduction of mitochondrial DNA with this treatment and measuring the type 1 interferon response. And what she found is with reduction of mitochondrial DNA, she sees a reduction of interferon response in a dose-dependent manner, suggesting that if not all, but partially, the mitochondrial DNA is responsible for tau-induced type 1 interferon response in microglia. So how about deleting cGAS? So this is what Joe did, is Joe took the cGAS knockout and the wild type and treated with tau and look at the signatures, the expression of the interferon, type 1 interferon signatures. So these are all the type 1 interferon genes expression, and you can see a dramatic reduction caused by cGAS knockout in the interferon response that stimulated by tau, showing that cGAS is required for type 1 interferon response in microglia treated with tau. How about in vivo? So we cross the cGAS knockout with the tauopathy mice and look at either mice with either 1 or 2 or 0 alleles of cGAS. And we perform a series experiment. And in this case, I'm showing you the single nuclear RNA-seq of microglia. And you can see that the all interferon stimulated genes are enhanced in the tauopathy mice and reduced by removing even 1 allele of cGAS, showing yes, in vivo, cGAS is also partially responsible for the type 1 interferon response in tauopathy mice. So we actually did not see any significant changes in the tau pathology per se. Instead, what we see is that at the same level of tau pathology, the synaptic integrity is protected by reducing or ablating cGAS. So here shows the immunoactivity of PSD95 in the CA1. It is reduced in the tauopathy mice, and this is partially ameliorated by removing 1 allele and more protected by removing 2 alleles of cGAS. And this resilience to the same level of tau pathology can also be shown with functional assays, and that including Morris-Waterman assay showing that the spatial learning is protected by removing 1 allele or both allele of cGAS. The only one that shows spatial learning deficit is the one that with normal cGAS with tau. And that is also true for spatial memory in the probe trial. The one that have no cGAS restored the preference to the target quadrant as similarly as those without tau. And in collaboration with Tara Tracy, Snap at Buck, we also examined the long-term potential in the perforant path circuit, and we find consistent with the previous finding that there is a reduction of LTP, especially the non-LTP in the tauopathy mice, and that is rescued by removing cGAS. So how does it work? So we were baffled until we performed single nuclear RNA-seq, and this data shows the genes that are changed in the excitatory neuron, as well as interneurons, driven by cGAS knockout on the tau background. And the gene that is called attention is a gene that Li Hui mentioned in her introduction, is MEF2C. So this gene called attention first because it is an AD risk gene. And it is known that the insufficiency of MAP2C can lead to intellectual disability, showing the importance of this gene in cognitive function. And the thing that excited us the most is a paper that Li Hui mentioned by Ravi and other colleagues, showing that a neuronal MAP2C can actually is a hub gene or is a driver to confer cognitive resilience in human AD brains that carry the same level of amyloid and tau. So that got us really excited. So we reached out to Ravi and Li Hui, and they kindly provided us with the MAP2C target genes. So we then overlay, Gloria, the graduate student, then overlaid the MAP2C target genes with the differentially expressed genes in either excitatory neurons or inhibitory neurons. And we looked at it statistically and unbiasedly about the enrichment. And we were very excited to find that the MAP2C target genes have a strong overlap with the genes that differentially regulated by cGAS deletion, suggesting that cGAS deletion markedly enhanced the transcriptional network of MAP2C. And one way to look at it is that a lot of the changes that caused by cGAS deletion in the excitatory and inhibitory neurons in the tauopathy mice can be largely driven or explained by enhanced MAP2C transcriptional network. So we then move on to try to move it into translational by treating the animals with a small molecule brain permeable cGAS inhibitor. So so this again was described in this published work. We established a PK of this small molecule and treated the PS19 mice after the onset of the disease at 6 months of age and treated for 3 months. We looked at memory. We also looked at synaptic plasticity. We found that the small molecule phenocopies the inhibition, the protective effect of genetic inhibition of cGAS, showing a restoration of the object preference by the treatment, as well as significant improvement of LTP. We also looked at the MAP2C target genes by looking at the single nuclear RNA-seq. Again, we find that the MAP2C target genes are significantly enriched in the DEGs that caused by the inhibition of cGAS. And again, we also find synaptic integrity is improved by the inhibition of the cGAS inhibitor, as shown with the PSD95 in neuroactivity, which was reduced in PS95, but restored by the treatment. So just to summarize the second vignette of showing you that the cGAS activation that could triggered by the tau in the AD model and other primary tauopathy models could drive the type 1 interferon response and MAP2C deficiency, and that could underlie the loss of cognitive resilience after years of accumulation of amyloid and tau. And inhibition of that could remove the hyperactivation of type 1 interferon response and enhance MAP2C and enhance cognitive resilience. So our working model is that by inhibiting cGAS and enhance the MAP2C transcription network, we can provide a new therapeutic paradigm to enhance the cognitive resilience. So with that, I thank the people along the way. And again, a shout out to Ravi and Li Hui for the collaboration and our funding agencies. And thank you for your attention.
Thank you, Professor Li Gan. You're too kind to us, and it's been fun to see the work progress so well. Anyone have any questions, feel free to raise your hand among the speakers, and if not, I will go to the chat box for any speaker questions. Go ahead, Li Wei.
Hi, Lee. Wonderful presentation. I really enjoyed it. The first part of your talk about the role of NF-kappa B in microglia and tau seeding and spread, as I understand it. So I'm always very curious to know how does this work? Like, how do microglia promote tau pathology? Do you have any thoughts about this?
Yeah, so that's very, very important question, and thanks for raising that. One of the working hypotheses, one we're working on, is how does tau processing in microglia get affected by NF-kappa B activation, and in particular, the proteolytic cleavage of tau in the lysosome get affected by NF-kappa B activation. So we have evidence suggesting that the retention of so actually it's it's in the paper as well. The retention of tau peptide or fibrils in the microglia is enhanced if NF-kappa B is inactivated, meaning less gets spared out, and also the form of tau is different with or without the hyperactivation of NF-kappa B. So I would say it's a processing, like the trafficking, as well as the proteolytic cleavage, most likely needed by lysosome. That is acting as a
There's a proteostasis problem in the microglia.
Yeah, so it's I would, if you use analogy, is that what gets spilled out by microglia is a better seed than the one that has not gone through microglia processing. So that's why if you have a more toxic seed that is more easy to be taken up by neurons to seed the as a template, and that process is driven by NF-kappa B activation.
Fascinating. Thank you.
Time for one last question. Elaine O'Loughlin in the audience is asking, do people with MEF2C mutations have impaired cGAS-STING interferon signaling? If this falls on excitatory and inhibitory neurons, is this relevant for tauopathies in, say, epilepsy or other neurological conditions?
Yeah, and thanks for asking that fascinating question. Actually, if you look at the MEF2C targets, many of those genes are epilepsy genes, including the sodium channel 1.1. So yes, the MEF2C hyperinsufficiency would lead to downregulation or would lead to regulation of the genes that could make those make the cells or neurons more excitable. So whether they have cGAS and interferon abnormality, we're not sure yet. But it could also be that it is downstream, the cGAS-STING activation, or MEF2C is downstream of the interferon response. So it won't have interferon signatures in the MEF2C hyperinsufficiency. That's also possible, but fascinating possibility that these two feed into each other.